got me? I'm already going on. Then, the second slide from that is just a brief description of the differences between the three types of muscle tissue. That's there. Looks like you're only gonna be on that one. I don't know if you guys wanna move over to that side of the room. Wish I couldn't get this to be more clear. Nobody fixes shit around here. Okay, just remember that skeletal muscle are few cells, dozens to hundreds of individual cells fused to form a skeletal muscle fiber. That's why you have peripheral nuclei. All the individual nuclei for those dozens to hundreds just get shoved off to the side. It has obvious striations, and that's because all the sarcomeres line up across that muscle fiber. Both smooth and cardiac muscles are unfused. They are individual cells. Smooth muscle cells are spindle-shaped, require hormones or autonomic control. Some can actually contract just by stretch. Cardiac muscle is also individual cells. If you didn't get an exam 3 study guide, I got some up here. It has sarcomeres, but just not as dense as skeletal muscle, and so it has subtle striations. The cardiac muscle works as a single unit because of things like intercalated disc and cell branching called anastomoses. Both smooth and cardiac muscle is involuntary only. Skeletal muscle is both voluntary and involuntary. Okay? So where it says voluntary movement, right here, draw a line off to the side and write mostly voluntary movement. Skeletal muscle is both voluntary and involuntary. So from your textbook, just a reminder of the anatomical differences. Here's something that is probably pretty important to remember for this section of physiology, as well as for a TEAS test. You should be able to go from big to small or small to big on a Scantron question. Let's go from big to small. The muscle, whatever muscle it is the organ of the skeletal muscle organ system. Within that organ, the muscle fibers will run in bundles. The name of the bundles is fascicle, so I would write fascicle right here. An individual muscle fiber right here, I would write the cell, as it says right here. Remember that the fiber is the skeletal muscle cell. And the reason why it is considered the cell, even though dozens to hundreds fuse to form it, is because it has an intact plasma membrane. Because it has an intact plasma membrane, even though it's huge, we call that the cell. Within the muscle fiber, there are columns of contractile proteins. Those are called myofibrils. And within the myofibrils, we'll have two different types of myofilaments, thick and thin. And how they are anchored on these Z lines produces these repeating patterns we call sarcomeres. And the reality is all the myofibrils in that cell are going to line up Z lines. And if you line up all the Z lines, you have obvious striations. That's why skeletal muscle is called striated muscle. Obvious stripes, because all the Z lines line up. Are we clear? And if you want to know what I just covered, it's up here. Muscle, fascicle, fiber, myofibrils, myofilaments, sarcomeres. Okay? You should also be able to go from small to big, so it can go in the reverse order. Everybody got it? This is all review of anatomy, isn't it? But we have to understand the anatomy to talk about our sliding filament theory. Okay, let's talk about some of the sarco words that are here on this figure. You do need to know this. The sarcomere is the smallest contractile unit of skeletal muscle. It goes from Z line to Z line. I've drawn Z lines here in green. So this is a sarcomere, and when a muscle contracts, the Z lines actually come closer together. Actually, you're going to have to learn that that is isotonic contraction. This is not something we talked about in anatomy. With an isotonic contraction, the Z lines actually get closer together. And sarcoplasm is just the cytoplasm. The reason why we use these words is because this cell is freakishly large, because dozens to hundreds of individual cells fuse to form it. But the reason why we call the fiber, muscle fiber, the cell is because it has an intact sarcolemma. That's the intact plasma membrane. It has some extensions of the sarcolemma that go deep, because not only are these fibers long, they're also thick and girthy. Everybody get what I just drew? Green line is the sarcolemma, but these little tubules that go deep into the thick muscle fiber is called the T-tubules. That's short for transverse tubules. Then you will have these collections, these bags of smooth endoplasmic reticulum. Smooth endoplasmic reticulum in a skeletal muscle fiber is called sarcoplasmic reticulum. In other cells, it's making lipids and carbohydrates and other things. In skeletal muscle, it does one thing, and that stores and releases calcium ion. That's what the smooth ER does. In skeletal muscle, we call it sarcoplasmic reticulum. But next to the T-tubule, there's going to be a collection on either side of the T-tubule called terminal cisternae. This is a collection of smooth endoplasmic reticulum right next to the T-tubule. Everybody, does this all look familiar from anatomy? Mm-hmm. So you have 1, 2, 3. Terminal cisterna, T-tubule, terminal cisterna. Everybody see the 1, 2, 3? That's why we call these triads. 1, 2, 3. Okay? And that's what you see here. The green T-tubule and a bag of smooth endoplasmic reticulum we call sarcoplasmic reticulum on either side. It doesn't label the terminal cisterna, but that bag next to the T-tubule is a terminal cisterna. Here's one of them right here. Doesn't that kind of look like what I drew? Mm-hmm. Any questions? You got to know these words. All right, let's talk about the sarcomere. Go ahead and fill in the blanks here. Maybe we'll just have You depend on the textbook and my PowerPoints for exams for the rest of the semester.
No.
And the severe introvert can enjoy not being in front of classrooms. There you go. All the information's there. Know all of it. Online exam. Good luck to you. That'd be awesome. I'll get a lot of bass practicing. Hey, you made it.
Sorry.
By the way, the owners of Harry's and Pismo has got pissed off at the manager because the manager didn't book us enough gigs in 2012. And so he's making the manager book us for the Pismo car show weekend in Grand Headline.
I was running. Sorry.
I'm too old for this. Okay, you guys ready? Let's go over the sarcomere.
Let's do it.
What you want to write next to this slide is where you have both types of myofilaments, it's dark. Where you only have one type of myofilament, it's light. Let me say that again since I didn't see everybody writing this down. Where you have both types of myofilaments, it's dark. Where you only have one type of myofilament, it's light. So, the anisotropic band, or A band, has both, so it is dark. It has both the red ones and the greens ones here, so we call this the A band. Both types, it's dark. I band make this symmetrical, but oh well. You only have the thin myofilaments. There's thick and thin. This only has thin. Because of it, it's light. That's the isotropic band. Thin filaments. The Z line runs right in the middle of it. So an I band actually involves two sarcomeres. Two adjacent sarcomeres. Third bullet, Z line. It says it there, but you better make sure you understand what it means, that word in the third bullet. The Z line is the foundational anchor to the thin filaments. All these thin filaments are tied to the Z line. Now when the muscle is stretched, right in the middle of the A bands, you actually have a little zone which only has thick filaments. And again, if it only has one type, it's light. We call that the H zone. Only has thick, so it's light. You only see it when the muscle is relaxed or stretched. When it's fully contracted, this disappears. Everybody got it? Does this sound familiar?
Yeah.
Now what you should write down here where it says M line, the M line is the foundational anchor to the thick filaments, as I've drawn it. I think you guys will be better students if we don't have class anymore and you have to take the test just off the textbook and the PowerPoint.
Oh my gosh, you're crazy.
It'll make you guys better, stronger students and not dependent on me to do all these multicolored drawings on the whiteboard. Yes?
Can you repeat again what the Z line is the foundation?
It is the foundational anchor to the thin filaments. All the thin filaments are anchored to it. All the thick filaments are anchored to the M line. God, you guys would be so much better off.
No.
You'll learn this shit on your own.
Your drawings are anchored to my grade.
All right, any questions? All right, now you know the sarcomere. And again, all the myofibrils in this muscle fiber are all going to sync up Z lines. That's why skeletal muscle fibers have obvious stripes or striations.
Okay.
You can see in the middle of the A band is the light H zone if the muscle is relaxed or stretched. In the middle of it is the M line. Just kind of how I drew it there, right? All right, let's talk about the thick and thin filaments. Thick filaments really only have one thing, and that's myosin. There's another protein that ties it all together to the M line. But don't worry about that. The thick filament is made up of these myosin proteins. If you look carefully at a myosin protein, it looks like a golf club. Doesn't it?
Yeah.
Except this golf club head swivels. Notice that all the golf clubs are arranged in a smooth helix or spiral. Do you see that?
Mm-hmm.
All the shafts of all those golf clubs make this filament thick. Do you see that?
Mm-hmm.
Any questions about the thick filament? Thin filament has 3 proteins. And it has a double helix of G-actin pearls. Each, and if you look at a G-actin pearl, it looks like a golf ball. Doesn't that make sense? You got a golf club, you get golf balls. So you have a double helix of golf balls, G-actin pearls. Each G-actin pearl has a binding site for a myosin head. There is a rope-like protein that blocks those binding sites. The binding sites smoothly go in also a double helix. Because it is a double helix, you need 2 ropes, and the name of the ropes are tropomyosin. The ropes are held in place by a snowman-looking protein. Actually, there are 3 proteins. That's why it looks like a snowman. And because there are 3 proteins, it's called troponin complex. It's not just a single protein. It's 3 of them. Everybody see that? Everybody see the snowman?
Yeah.
This snowman can do sit-ups. So you have to have these 3 proteins in mind when you're talking about the thin filament. Okay, everybody got this? Here's the double helix of G-actin pearls. Double helix. Each black spot is the binding site. Normally it is blocked by 2 ropes called tropomyosin, and the ropes that are held in place by the snowman troponin complex. This snowman can do sit-ups. Okay. Here we go. My anatomy students, does this kind of look a little familiar?
Yeah.
Don't take it for granted, this is more detailed. But if you took me for anatomy, you got 80% of this already. We're gonna do it in 10 steps. One thing I want you to get out of this figure on the left is that when there is isotonic contraction, and that means there's sliding film. This is all about isotonic contractions. We're going to learn later about isometric contractions, and in isometric contractions, there is no sliding filaments. So if you're talking sliding filaments, you're talking about isotonic contractions. And when the filaments are sliding, the Z lines come closer together, the muscle shortens. In a nutshell, this is isotonic contraction. Do you see my muscle shortening? In other words, there's movement. Everybody got it? This is isometric contraction. Do you see any movement?
Mm-mm.
There's still power strokes and cross bridging, there's just no sliding. Because the tension can't overcome the load, we'll talk about that later. But when you have isotonic contraction, notice that the A band, and this is what I want you to get, the picture on the left, A band does not change. And the reason why is the A band is defined by the length of the thick filaments. Do you see where it's labeled A band? You see how it is the length of the thick filaments, and the length of the thick filaments do not change.
Right.
So, when the muscle does isotonic contraction, length of the thick filaments don't change, the A band doesn't change. What disappears is the I band and the H zone. Do you see that in that picture?
Yeah.
You better know that. A band stays the same, I band and H zone disappear. You can see this on this animation on the bottom right. A band stays the same, I band and H zone disappear. This is why I say you only see that H zone when the muscle is relaxed or stretched. If it's fully contracted, it's gone. Any questions? All right. You have to start with the synapse from the motor neuron. The motor end plate is the axon terminal. It's going to get really close to the sarcolemma. Motor end plate plus sarcolemma equals the neuromuscular junction. Now, because we talked about synapses, I should be able to do this fast. In the motor end plate are vesicles of acetylcholine. Here's part of the 20%, my anatomy students, that wasn't in this in anatomy. You also have to know what a motor unit is. A motor unit is a single motor neuron. And remember, neurons branch telodendria. A single motor neuron and all the skeletal muscle fibers it innervates. That's a motor unit.
Okay.
And they are different. The ones on your face and in your fingers are really small. You can't generate a lot of strength because they are small motor units, but you have great coordination. You could do surgery with these small motor units. You following me?
Mm-hmm.
With gluteus maximus, you've got these huge motor units, hundreds and hundreds of fibers to one motor neuron.
Yeah.
That'll help you get out of the blocks in track with your sprinting. Great force. But I don't know anybody who can do surgery with their ass. Does this make sense?
Yeah.
Okay, you do have to know what that is. That's part of, oh, and by the way, I'll have a handout for the essays for lecture exam 3.
Mm-hmm.
This sequence is going to be split up into 3 different essays.
So are you saying that on your ass you have more of these, or are they larger?
They're larger.
Okay, sorry.
There's more fibers per motor neuron.
Okay.
So you can generate great strength without much fine motor control.
Okay.
Alright. And this figure from your textbook shows 3 motor units. Everybody got it? We're going to talk later about different types of skeletal muscle fibers. A motor unit never shares. It's only for one type of skeletal muscle fiber.
Okay.
Okay? All right. Well, when the action potential reaches the motor end plate, it triggers exocytosis. Acetylcholine is released, crosses the synaptic cleft, and binds to acetylcholine receptors on the sarcolemma. These little insignias here, steps 1 through 3, this has to do with the long sequence on page 41 in your lecture packet. That's just too many steps. 20. So I combined them in the PowerPoint. Okay? All right, so now we got acetylcholine on acetylcholine receptors on the sarcolemma. That's gonna cause there to be a huge influx of sodium, very little potassium leaks out, you can see it here. That's depolarizing. This animation, I had to change this, anyway, long story, this animation doesn't work. And in the newer DVDs the publisher gave out, they excluded this one, which was a bummer, because this was really good to have the whole sequence here. But let's go on. When you have depolarizing of the sarcolemma, you're gonna get a new action potential that's gonna spread down the entire sarcolemma, and it goes down the T-tubules. It follows the green line. What happens then? That action potential is gonna cause the calcium pumps to turn off on the sarcoplasmic reticulum, and also opens up calcium gates, allowing this calcium to be released out of the sarcoplasmic reticulum. It's gonna be released from the sarcoplasmic reticulum to the sarcoplasm. Do you see why we gotta learn all these sarco words?
Mm-hmm.
What happens when there's calcium in the sarcoplasm? Well, you gotta remember what the thin filament looked like. And the thin filament had those two ropes blocking the binding sites, held in place by troponin. When calcium is released, it binds to the snowman. Troponin, it does a sit-up. And when it changes shape, it moves the rope off the binding sites. Tropomyosin is now off the binding sites. That automatically results in a cross bridge because these guys are really close together.
Mm-hmm.
Everybody got this? What happens when there's a cross bridge? It automatically is gonna power stroke. This is just a figure showing you what we've been talking about so far. This whole thing is referred to as excitation contraction coupling. I just fixed this PowerPoint before lecturing. That's, hopefully this thing works.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
that good?
Yeah, it's good.
Man, see, you guys are just going to textbook.
No.
Typically, a single motor neuron arising in the brain
All right, why does it result in a power stroke? It results in a the cross bridge results in a power stroke because there is stored energy. Key here is from a previous ATP. A previous ATP had already donated its energy and it caught the myosin head in an energized form. You need a new ATP to detach the cross bridge. But this caught position was from a previous ATP. Everybody got it?
Mm-hmm.
And then you gotta recognize the cross bridge cycle, these repeated cycles of 4. This animation kind of goes really fast. But let's go over this animation, even though it goes really fast, at least it keeps cycling. Does everybody see the double helix of golf balls? GIAC and pearls in yellow.
Yeah.
Everybody see the 2 ropes in blue, tropomyosin.
Yes.
Everybody see the purple snowman, troponin.
Yep.
Everybody see how there is a yellow ball that binds to the snowman.
Mm-hmm.
That's calcium ion.
Mm-hmm.
When the calcium ion binds to troponin, it does a sit-up. Everybody see it doing a sit-up?
Yep.
When it does a sit-up, it moves the rope, tropomyosin, off the binding site. When it does that, it results in a cross bridge, and you can see the golf club looking myosin head binding to a golf ball. When it binds, it automatically does a power stroke. The ATP is the green ball. You can see that with magnesium, well, that's on the next slide, it hydrolyzes the ATP to ADP and P. Everybody see that? But it is a previous hydrolysis that cocks the myosin head. You need a new green ball to dislodge the cross bridge or detach a cross bridge, and that's happening here, 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 here. Everybody see that? The sequence is cross bridge, power stroke, detachment, re-energizing or recocking. Cross bridge, power stroke, detachment, recocking. Cross bridge, power stroke, detachment, recocking. Cross bridge, power stroke, detachment, recocking. And the really good illustration of this would be in the Summer Olympics, but who knows if Japan's gonna have Summer Olympics now. It's in Olympic rowing. Same sequence of 4. The filament that's sliding is the boat. They put the oars in the water, cross bridge. They power stroke. They take the oars out, attachment, reposition, recocking. Same sequence of 4. Cross bridge, power stroke, detachment, recocking. Cross bridge, power stroke, detachment, recocking. And you got a sliding boat. Everybody got it?
Yeah.
If you watch Olympic rowing with a critical eye, you will see that the oars actually don't move in the water. When these athletes do the power stroke, the only thing that moves is the boat, not the oars. It's like the oars are going into mud. They don't move in the water.
Impressive.
Okay? All right, now this is a little bit more than 20% that's not covered in anatomy. Oh, let's first play this video. This is the cycle.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules in thin myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross-bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin binding site on actin.
Any questions? Cross bridge, power stroke, detachment, retie. Okay? And again, this only applies to isotonic contractions. That's where the muscle actually physically shortens and there's movement. Are we gonna have to reset the whole thing?
The contraction of the skeletal.
This is the new information now, muscle and anatomy. So we're using the analogy of a golf club for the myosin. What you should know is that there are 3 different types of golf clubs. Okay, so they all a little bit different. We call those ATP, myosin ATPase isoenzymes.
All 3 of them? That's what you call them?
Yeah, they're all different forms of
that.
Yeah, so the myosin head is actually an enzyme.
Okay.
and it's myosin ATPase, and there's 3 different subforms of them. Those are isoenzymes, and that's why there's 3 different types of skeletal muscle fibers. Okay?
Okay.
Any questions on this? By the way, this explains what happens when a body dies. When a body dies, not all the tissues and organs die at the same time. It actually takes hours for a body to actually completely die unless it's destroyed. If someone just passes away, organ systems are dying at different times, and it takes hours. It takes a long time to get the food done. Everybody got it?
Yeah.
But one of the things that does happen, and it's well known, is that when the body dies, it gets stiff. And that's because it's still doing these cross-bridge and power strokes. But when it runs out of ATP, it can no longer do the 3rd step in the cycle. Cross-bridge power stroke, no new ATP for detachment. And you've got myosin head still bound to the thin filament. That's why bodies get stiff in rigor mortis. Does that make sense?
Mm-hmm.
Eventually, the filaments start falling apart, and that's why, like in our cadavers, we can actually, they're pretty stiff because they're cold. But actually, we can actually move things around, but we're ripping up filaments when we're doing it. And myosin heads are still connected to the thin filaments. Does this make sense?
Yeah.
All right, so that's contraction. Steps 1 through 5 is contraction. 6 through 10 is how to shut it off. You guys remember when we were talking about drug effects at synapses?
Mm-hmm.
And we were talking about how a lot of those toxins kill you by suffocation?
Mm-hmm.
Everybody look at me. Some kill you by suffocation because you can't contract the skeletal muscle, right? Like rattlesnake poison, crotoxin. Some kill you by suffocation because you can't turn your muscles off.
Mm-hmm.
Then you can't exhale. If you can't exhale, you can't keep inhaling. Everybody got it?
Yeah.
There's 2 ways of being suffocated at synapses. One interferes with steps 1 through 5, one way. The other way is interfere with steps 6 through 10. You're dead either way. It's just a different way of killing it. Does this make sense? All right. So you got to turn off the action potential. This is done by acetylcholinesterase. It's actually in the synaptic cleft, and it breaks apart acetylcholine into 2-carbon acetyl group and choline. The action potential ends. Once there's no more action potentials going on the sarcolemma, the calcium pumps and sarcoplasmic reticulum turn back on, and they pump the calcium back into the bag, sarcoplasmic reticulum. When the levels of calcium ion and the sarcoplasm goes back to resting levels, it falls off the snowman. When it falls off the snowman sits back, lays back down. When it lays back down, it moves the rope, tropomyosin, back over the actin binding sites, blocking any cross bridges. No more cross bridges, no more power stroke, contractions ends. And here's something else. The muscle can passively return to resting length or actively pull to resting length by a muscle antagonist. Do you guys remember what muscle antagonists are?
Yeah.
So if you do a muscle contraction with the biceps brachii, you can either just relax it, or you can actively pull it with the triceps brachii.
Mm-hmm.
Everybody got it?
Mm-hmm.
We lost both screens now. TB working, we can try to fix the rest of this after I'm done. Hey, this could be a trial run for COVID-19. This last is over, and you guys can just leave it in the textbook.
No. No way. I'd rather get the coronavirus.
You guys are gonna become much stronger students.
I think we're offering, like, people to volunteer money, if they volunteer to, like, be tested, will be infected with the virus, and then make a vaccine, like $7,000 or something.
Yeah, it still is gonna take a long time. It's gonna take at the soonest to the end of this year. Because not only do you have, you can actually make it that way, you gotta refine it. And then you gotta test it. And then you gotta mass produce it. And then you gotta deliver it to every corner on the planet. There's only one continent that doesn't have it, and that's Antarctica.
Oh my god. I'm moving
It
there.
will take months. So, but yeah, the more anti-COVID-19 they can get, the faster the testing and mass production can go. You know how they'll probably make the antibody? Find these files. They'll find the gene to make it, insert the gene in the cow, make a whole bunch of cows make anti-COVID-19. You gotta refine it all. It'll be human, it's a human protein, but it's just an animal something. Well, we got the one screen, we better move on or else I'm not gonna finish. Yeah, maybe that might be good, let's just go with the one screen, and we'll finish this, you can finish this after. Yeah, you can, I can get around. Thanks. All right, let's talk about what happens to a skeletal muscle fiber during exercise. Just to give you some context, when a normal cell doubles the rate it's burning ATP, that is a cell that's working its ass off. Not that often do cells double their rate of ATP usage. Now talk about skeletal muscle. Between resting and exercise, it could increase the rate that it's burning ATP 100-fold. This is a completely different league to other cells. Everybody got it? So on this one here, you want to go ahead and turn to page 44. Now, in lecture exam 1, we covered aerobic and anaerobic respirations, ways of making ATP. Do these figures look familiar from that lecture exam 1 material? They're the same figures. Everybody, does that look familiar?
I see it.
Well, because muscle cells have such a high demand, there is a third pathway that other cells don't have, and that's the dephosphorylation of creatine phosphate. And as soon as you start contracting muscle, this gets burned first. basically creatinine phosphate. It has a high energy phosphate group on it. It just donates to ADP. Okay? It's really quick. But this is only like 10 to 20 seconds. Then you hit the fork in the road. Then it's all about what kind of shape you're in and what kind of exercise are you doing. If you're a couch potato, or even if you're a fine-tuned athlete and you're doing intense exercise, it's going to do this. And it's going to go to anaerobic respiration. But you're only getting 2 ATP per glucose. You can only use glucose. You're going to generate lactic acid. You're going to feel the burn. And this is like 30 to 90 seconds. Most people, it's most like 30 seconds. World-class athletes can do this for about 90 seconds. Okay? But it's painful. Again, I don't know if we're going to have the Olympics this year, but in track, watch the dudes that are doing the 800. They're sprinting 2 laps around the track. About 90 seconds, they cross the finish line, and they are looking hurting. Their bodies are on fire from lactic acid. Okay, so we got this figure here with 1, 2, and 3. This is not the order that they happen. I'm covering 1 and 2 first because we covered that in lecture exam 1. 3 is the one I'm adding right now. So on this page 44, let's see if this will work. You can write these numbers down that correspond to this slide here, 61. Aerobic respiration is number 1, that's with oxygen. Anaerobic respiration is number 2, without oxygen, yielding lactic acid. And the dephosphorylation of creatinine phosphate is 3. That's more like the order with which you learned it, right? We covered aerobic respiration, anaerobic respiration at the same time, and then now with the creatinine phosphate. Okay. So now let's talk about the fork in the road. So this is what it's actually is in real life. First, it's this. When it's depleted, then you hit the fork in the road. Now we're going to talk about the fork in the road. Everybody understand that? Mm-hmm. Because this is almost indefinite. Almost. Aerobic respiration, you must have oxygen, but you can use all 3 nutrients. You can use protein if you have to. Anaerobic respiration, you can only use glucose. Remember, you get the big payoff in the electron transport chain, and you get 36 ATP for glucose, and that's why this is almost indefinite. You have plenty of ATP. For anaerobic respiration, you're only having 2, and then you're building up lactic acid, which is why you can't do that for very long. And this is showing you the factors of what causes which fork you go in. Aerobic respiration, you got to be in shape, and you have to do a moderate type of exercise. And and then it can last almost indefinitely. Anaerobic respiration, either couch potato, or you can even be a world-class athlete, but if you do anything intense, sprints, powerlifting, you're doing anaerobic respiration. And again, you're normally looking at 30 to 90 seconds. Like those pictures?
Is that an actual potato?
Yeah.
Oh my god.
So everybody knows what the actual order is now? It's this first, then a fork in the road. Okay, go ahead and fill in the blanks and let me explain this. We'll talk about this too in lab 9, second week after spring break, if we are still holding classes. Yeah, I don't know what they're gonna do about labs if they close campus. I think there is a serious possibility you guys might all have to take incompletes and finish this in a future semester. Because there's no way to do labs remotely.
Can we just be quarantined with you?
Right, wear masks and stuff. I'll buy it.
Yeah, just save the mask for when you're in your car by yourself. All right, oxygen debt it's a real thing, but what you need to know about oxygen debt is that it's really not what your body is about. The reason why is that oxygen does not affect pH. CO2 affects pH because of this. Most of your body is water. Plasma is definitely mostly water. You put CO2 into it, it becomes this, carbonic acid, which will dissociate into this plus bicarbonate ion. This right here is the H of pH. That's acid. You add, this is an equilibrium, that's what these double-sided arrows mean. It's an equilibrium, it wants to stay level. If you increase CO2, you're gonna increase acidity. You blow off CO2, you make yourself alkaline. Everybody got it? While oxygen does not affect pH, it doesn't really affect your respiratory centers in your brain unless you get really low in oxygen, and I mean really low. Minus 60 millimeters partial pressure of oxygen. If it's, and that's really low. If you're not at that level, your body doesn't even respond to oxygen. So even though we're talking about oxygen jet, which is a real thing, that's not what triggers you to breathe hard. What triggers you to breathe hard is acidity. And you know that if you eat a lot of spicy food, by the end of that meal, you're doing this. Because that is, all that spicy hot food is acidic. Then you can actually eat enough to start making your pH, body pH decrease. Remember, we are centered around 7.4. If you get to 7 even, neutral, you feel like absolute shit, like you want to die. You do not want to be neutral. But you get to 7.25, you're already, why? I'm blowing off CO2. Remember that equilibrium. You blow off CO2, you get rid of acid. Everybody got it? And oh, by the way, if you're living on this planet, if you're blowing off CO2 out into the atmosphere, you're also sucking up oxygen. So even if oxygen debt is real, that's not what triggers your breathing. What triggers your breathing is acidity from both too much carbon dioxide and lactic acid. Both of those are gonna make you breathe hard. And when you breathe hard, you're gonna get more oxygen. Everybody understand how the body works? All right. Fatigue is three things. Buildup of lactic acid, what's called lactic acidosis. 2, ADP depletion, because you're only getting 2 per glucose. And 3, electrolyte imbalance. That's where it's talking about calcium and potassium. Those are the 3 things. Lactic acid buildup, depletion of ATP, electrolyte imbalance. Electrolyte imbalance is the biochemistry of Gatorade. It was actually done by physiologists. Did you guys even know that? All right, what happens when you exercise? Well, you gotta feel the burn. The burn is actually a good thing. The burn for your body is the same as a headache from studying. It is a good thing because it's causing chemical signals in your body to physically change you. We already talked about what happens in your hippocampus when you make yourself remember more. Well, this is now making your body lift more or run farther or run faster. You're going to feel the burn. If you feel it enough times a week for enough weeks in a row, the mitochondria actually start growing. When they get up to a certain size, their ring-like DNA replicates. Then that mitochondria is going to divide. You started out with 1, you now have 2. You keep doing that, and those 2 are going to get bigger until they reach a certain point, and they go to 4. You keep working out and feeling the burn, those 4 will go to 8. Everybody got what you're going to do to mitochondria?
Yeah.
Endosymbiosis. Also, your sarcoplasm is going to build up stores of myoglobin. Your muscle cells will actually be able to store more oxygen. You're going to see angiogenesis, first to the skeletal muscle to be able to deliver more oxygen. And once it can get oxygen, now the weak link is nutrients. You will actually get angiogenesis to your adipose. Your body becomes a fat-sucking machine.
Ooh. Yeah.
I mean, I'm freaking old. Did you guys know I graduated from college in the 70s? Do the math.
College?
Uh-huh.
Okay.
I also graduated from middle school in the 70s.
Wait, what? Really?
Yeah.
Okay.
I didn't go fast.
Oh, 10 years?
Yeah.
Yeah.
71 I graduated from middle school. 75 I graduated from high school. 79 at age 21 I got my bachelor's. I got my college degree in the 70s. Because of this knowledge, again, you are learning about you. It affects my choices. Now that I know this, I spend all day sucking off my fat. I spend all night trying to put it back.
Yeah.
I'm an old person who doesn't watch what he eats because I do this. Everybody got it?
Yeah.
All right. Also, you know, once skeletal muscle fibers are formed, once those cells fuse, they no longer can go through IPMAT, no more cell cycles. You have all the skeletal muscle fibers you'll ever have. So if you want to bulk up, you're not increasing the number of muscle fibers you have, you're just making the muscle fibers you have bigger by filling them up with myofibrils. Everybody got it? Mm-hmm. And then you can get huge. Don't look at this picture too close because I think she's got something in her shorts.
Oh my God.
That'll be your father leaving you for spring break. All right, so again, lecture exam 2 is going to be the second Monday once we get back. Okay, not the first day we're back, a week from the first day we're back.
Yeah.
Spring break.
spring break now?